All right. And did you discuss starting recording this?
Yes.
Okay.
Yeah.
Yeah. Mm-hmm. So your your 1.2 is, when are you going to get solid state probes for it though?
Will take several months from now.
Uh-huh. Oh no, they, I mean, I think in Göttingen and in and clearly in Zurich, but in Göttingen, they're doing a lot of solid state. I mean, the liquids probes they had turned out not to be very good, at least initially. I think they're okay now, but yeah.
In Utrecht, I think they also getting a solid state, or they got it already. I'm not sure.
Mm-hmm. Yeah.
So how many people from your lab, Rob, are attending the seminar?
Oh, basically everyone. And in Mahong's lab. It
Which
varies. And then I've had 3 students just, graduate recently. So 2 of them are working a lot on amyloid. One of them's in Utrecht right now with Mark. She and her husband's a professor at Amsterdam, a computer scientist. And yeah. And the other one just left last week. So, but it, as I said, it varies a lot from week to week, right? Yeah. So how are things in Japan? Are they, I mean, is the COVID problem okay?
Well,
control?
not so bad, but there's a little restriction, like the restaurant closes early and occasionally elementary school closes.
Mm-hmm.
But life is not very bad compared to it used to be 1 year ago or 2 years ago.
Yeah. Mm-hmm. Mm-hmm. Yeah.
So hopefully it's getting better and better. Hopefully.
Yeah. Yeah. Well, as more people have been vaccinated and more people have had the disease, developed immunity. Yeah.
Yeah, I myself am going to be vaccinated this Friday. This is going to be my 3rd time.
Mm-hmm. Yeah. Well,
let's see.
Yeah. Mm-hmm. Yeah. No, that's good. Yeah. Mm-hmm. Yeah. Okay, another minute or 2, and we'll get going, right?
So what's the percentage of people vaccinated with at least 2 injections in Japan?
Oh, pardon, I'll say it again. I couldn't catch your word.
What's the percentage of vaccinated people in Japan, at least with 2
Wow,
doses?
at least 2. Almost everybody. I'm not informed, or I have to search the internet, but perhaps my guess is about 90% of people have already get vaccinated. I think it may not be correct.
Mm-hmm. Well, that's very good. That's quite high.
Yeah, it's similar in Italy. It's about 90% with 2 doses, and out of them, 70%, 60-70% as the 3rd booster.
Mm-hmm. Okay, so why don't we get going now? All right, so today we have two excellent talks, very different perspectives of magnetic resonance. The first talk is by Kazuyuki Takeda, who is from Kyoto University in Japan, the Faculty of Science. And the second talk is Claudio Luchinat, who is from Florence, University of Florence in Italy. And he will be telling us about mixed fibrils of A beta amyloids, okay? And Takeda's talk is going to be about upconversion of radio frequency NMR signals to light via a membrane transducer. So he's going to tell us how to do optical detection of magnetic resonance, so to speak, right? Okay, so why don't you go ahead, Takeda, with your presentation.
Okay, thank you very much for kind introduction, Bob. And I thank Bob and Kong for kind invitation and arrangement of this seminar. And may I share the screen now? I already practiced just a while ago, so I hope it works. Again, I hope you can see the screen and the pointer.
Yeah, it's fine.
Okay, so today let me talk about what we call electromechano-optical NMR, or for brevity EMO NMR, that we put forth some time ago. And this slide describes a simple view of what it is. We employ an LC resonance circuit for the detector of magnetic resonance using a normal coil and somewhat exotic capacitor made of an elastic membrane oscillator. Then the electromotive force due to nuclear induction causes Coulomb force between the electrode of the capacitor, affecting the mechanical motion of the membrane. And in turn, the membrane dynamics is detected through an optical means. At least from the theoretical point of view, it was suggested that the ultimate attainable sensitivity exceeds that of the conventional electrical detection scheme. And to realize it, we use a commercially available silicon nitride membrane with a typical lateral size of 500 micrometers and typical thickness of about 50 nanometers, which is supported by a 5 by 5 millimeter silicon frame. A metal layer is made on the membrane by vacuum deposition. In addition, the counter electrode of the capacitor is made on a silica plate, and these parts are gently piled together to form the capacitor. As you can imagine, the membrane is a little fragile, but eventually we made something that worked. To avoid air friction and damping, the oscillator is put in vacuum. At first, we have made a somewhat massive cubic chamber equipped with a pair of hermetically sealed electrical ports and an optical window. And the metal layer made on the membrane also served for one of two mirrors of an optical cavity. And by inspecting the optical signal reflected back from the cavity, and we could observe some modes of characteristic oscillation of the membrane that reflect Brownian motion. The fundamental mode appeared at around 180 kilohertz, and at higher frequencies, 1, 2, or 2, 1, and 2, 2 modes, and so on were visible. And now the characteristic frequency of the mechanical oscillator appeared at sub-megahertz region, whereas the NMR frequency of interest is on the order of, tens of megahertz or hundreds of megahertz. To compensate such large mismatch in the characteristic frequency of the mechanical and electrical systems, a drive signal is required with a frequency given by either the sum or the difference of these characteristic frequencies. Then the two separate systems are allowed to communicate with each other, passing signals. And the diagram of the experimental setup looks like this. A solenoid coil dedicated for signal detection forms an LC resonant circuit with a membrane capacitor sitting in vacuum, whereas pulse excitation is performed with a separate saddle coil tuned at the same frequency. During signal acquisition, the drive signal is applied through the port shown on the left, and the optical signal reflected back from the optical cavity is detected and quadrilateral demodulated into the in-phase and the quadrilateral components. After somewhat desperate struggle, unsuccessful attempts, we finally managed to detect the first signal of EMO NMR using a desktop permanent 1 Tesla magnet at around 42 megahertz. And we were so happy about the successful proof of principle demonstration. However, the signal to noise ratio was terrible. Indeed, it was orders of magnitude worse compared to the conventional detection. And by changing the metal from gold to aluminum, and thereby by lightweighting the oscillator, the mechanical frequency increased and the signal to noise ratio got better, but still much lower than ideal. And we are now interested in making it at least better than that of electrically detected NMR, as it should be, provided that the system could be optimized. And now, let us consider a given ambient temperature of the circuit where Johnson noise develops. It's inevitable. And for the conventional NMR, the preamplifier used for signal amplification at the first stage faithfully amplifies the signal as well as the Johnson noise. And in addition, adds some extra noise. This is not avoidable. Whereas one interesting point in EMO NMR is that the signal is transduced through 2 steps, namely, firstly, from the electrical to the mechanical, and secondly, from the mechanical to the optical systems. Here, apart from the common Johnson noise, the major source of added noise is the Brownian motion of the mechanical oscillator. Now, if the system could be optimized to such an extent that the transduced Johnson noise overwhelms the Brownian noise, the effect of the latter would become insignificant. Toward this end, it is necessary to improve the efficiency of communication between the electrical and mechanical systems. Therefore, we wanted to make the system such that tiny displacement of the membrane changes the capacitance as much as possible. Even though such a requirement is challenging, it is at least a technical issue. And toward a straightforward solution, we decided to revise the design of the capacitor. In addition, we are interested in shifting from the desktop 1 Tesla magnet to a superconducting magnet used for modern NMR. That's why we started developing a palm top module for a part of an NMR probe that can be put inside the bore of the superconducting magnet. And this shows a top and the bottom view of the module containing the membrane capacitor. On top, there are a pair of hermetic ports connected to the membrane capacitor inside and to the cord outside the chamber body. And on the other side, you see an optical window and an evacuation port, a vacuum evacuation port. The metal-coated membrane and the counter electrode were piled together, put inside the chamber, and we designed the Gauss beam of the laser and arranged the required optical components accordingly. Now, a solenoid coil is connected to the membrane capacitor and tuned at 50 megahertz, corresponding to the carbon-13 resonance in a magnetic field of 4.7 Tesla. In addition, we installed another saddle coil. I hope you can see saddle coil doubly tuned at 250 megahertz, so that pulse excitation at proton and carbon-13 spins is possible in our 4.7 Tesla superconducting magnet. And we aimed at observing the carbon-13 induction signal by the EMO scheme. This is the circuit diagram. And the laser beam is applied through an optical fiber, a collimator, a convex cavity mirror onto the aluminum layer on the membrane sitting inside the vacuum chamber, palm-top vacuum chamber, and the light is reflected back into the fiber again and photodetected. And finally, quadrilateral demodulated into the magnetization components in a rotating frame. And we applied the drive signal to the circuit during signal acquisition, optical signal acquisition. And the drive signal was also used to generate a coherent reference signal at the characteristic frequency of the membrane oscillator by mixing it with the carbon-13 reference signal. The scene of the experiment looks like this, where you can see a laser device, optical fiber, photodetector, and vacuum pump, and so on. The vertical alignment of the optical pads inside the magnet bore and observation of the optical cavity by Yusuke Minaga, a PhD student, is a kind of miracle. Amazing. Good job. To demonstrate carbon-13 EMO NMO in combination with pulse techniques, we implemented refocused inept sequence with proton decoupling and managed to observe signals from liquid benzene. For comparison, the conventional NMR signals is also shown. Apparent difference in the noise profile is due to the difference in the bandwidths, detection bandwidths, and it is not so easy to make straightforward comparison of the sensitivity just by inspecting these data. Accordingly, we performed conversion of the synthesized CW RF signal to light and measured the power spectrum of the optical carrier. Here, the sharp spike here comes from the tone signal, whereas the broader skirt originates from the thermal or Brownian noise of the membrane oscillator. In this case, the height of the peak relative to the top of the heel of the Brownian noise measures the sensitivity. So honestly speaking, EMO is still behind the conventional electrical approach by 10 or 20 dB. It is a bit pity, but nevertheless shows good progress compared to our first demonstrations with the terrible sensitivity as shown earlier. One good news is the flow of the noise outside the region of Brownian noise is lower when we normalize the vertical scale with the tone peak height. So with some extra effort, I think we can go ahead of the electrical detection in terms of sensitivity eventually. It would also be fair to mention that the detection frequency bandwidth is currently around typically 100 hertz to at most a few kilohertz, you might find it unattractive. To increase the bandwidth without sacrificing the sensitivity, again, we need to strengthen the coupling between the electrical and mechanical systems. Then this profile broadens, making the bandwidths a bit larger without sacrificing sensitivity. On the contrary, the sensitivity would also be enhanced. So design of the capacitor, in particular, the gap between the electrodes matters. Meanwhile, we faced another challenge, namely heating of the membrane by laser. I mean, light absorbance of aluminum is just a few %, but the effect of heating is not negligible at all, partly because the pad is so tiny and thin. In addition, the channel of heat dissipation seems to be so weak, heating affected the optical parts slightly and thereby the characteristics of the optical cavity, making the transducer a bit unstable. So we no longer wanted to use the metal mirror, but still we do need the metal, capacitor electrode. Now, our idea was to spatially separate the mirror part and electrode part on a single membrane, utilizing a higher mode of characteristic oscillation of the membrane. We chose the 2-2 mode and to make a diagonal pair of metal capacitor and metal free mirror. And we came across with the idea of what is called metasurface, which is also known as photonic crystal slabs. The metasurface is composed of a periodic sub-wavelength structure showing interesting optical properties. Indeed, you can make a low-loss mirror, not by adding metal, but by subtracting the membrane material itself. Inspired by a group from an institute called LKB, located inside Sorbonne University in Paris, we started making a periodic array of nano-sized holes on the silicon nitride membrane. We owe a lot to wonderful people from joint project on quantum optics, optomechanics, gravitational wave detection, and magnetic resonance. The recipe is not so complicated. The membrane was resist coated first, and then the pattern was drawn with an electron beam, and then dry etched with CF4 plasma, and finally the resist was removed. Then you can make holes. And after some struggles, we managed to make a diagonal pair of metasurface mirrors on the membrane. And we used one of the two mirrors, leaving the other just unused. Then we made the spatially separated metasurface mirror and electrode. A student did a good job making an optical cavity with another mirror co-optical alignment and successfully observing 1-1 and the 2-2 modes of characteristic oscillation. And conversion of a synthesized RF signal to light was successful. And we found the new module with the metasurface was indeed robust or stable against laser beam irradiation compared to the previous aluminum mirror deposited onto the membrane. Let me summarize. Several years ago, we started the project, and it took a few years before EMO-NMO was experimentally realized for the first time. but the sensitivity was terrible. We have made some improvement, and there was apparent external silence for a few years. But the truth was we have been internally struggling with a lot of challenges. And recently we managed to make some more progress regarding the metal surface and demonstration of carbon-13 enamel in the superconducting magnet. Next thing to do for us is to go ahead of the conventional detection in terms of the signal to noise ratio for a given ambient temperature. Then we have some applications in mind to solid state enamel and MRI with some methodological modification to become compatible with the relatively narrow band EMO detector. One unique potential application would be mode cooling of the circuit, utilizing radiation pressure of the laser beam, which would allow us to get rid of thermal noise without decreasing the physical temperature in a normal sense. This is the most attractive application I find, even though I realize it's not easy at all. And finally, let me thank people who supported the project: Koji Usami, Kentaro Nagasaka, Yusuke Tominaga, and many, many other people. Without their efforts, no progress would have been made. I'm also grateful to funding sources. Okay, this is the end of my talk. Thank you for listening.
Okay, thank you very much. Very nice
Thank you.
presentation. Very interesting new way to detect magnetic resonance. And there's, I think, there's a comment in the chat from Mark Conradi. And he says, the force between the capacitor plates is quadratic in voltage. But for your transducer, you want a linear transduction. How do you accomplish this?
Well, at this moment, you're quite right. Very good point. And currently, the displacement is in a so-called very small region. So when we expand the displacement in Taylor series, yeah, we are working on a linear region. So if we reach the quadratic region, and this would mean that the transducer is saturating, just like amplifier saturates.
Okay, so why did you choose silicon nitride for your membranes as opposed to zirconia or diamond, for example? Okay.
Yeah, there are a lot of membrane devices in the world existing. I realize it. We realize it, and there are a lot of previous reports in the field of quantum mechanics and optomechanics where they reported the damping is the least. I don't know why, but it's reported that the damping is the least for the silicon nitride compared to other material. Yes, that's simply why we follow the previous reports.
Well, I mean, the thermal conductivity of diamond, for example, I mean, you have thermal conducting problems. Nitride, yeah. And the thermal conductivity of diamond is much better. I mean, and you can get very thin diamond sheets now, you know. Well, I'm not sure how thin, but I know they are available. Yeah.
Already, but some of its important point is not the thermal conductivity, but the mechanical dissipation of mechanical motion. It needs to be as small as possible.
Okay, so there's an anonymous attendee question. What is the theoretical limit for the signal noise improvement compared to classical detection?
Compared to classical electrical means, you
Yeah,
mean? Yeah. Okay.
Inductive detection, right.
Yeah, this is also an interesting point. And let's say the ultimate, yeah, we haven't reached to this point, but ultimately the efficiency enhancement corresponds to the conventional preamplifier with a noiseless or somewhat unrealistic noiseless preamplifier. I mean the noise figure of 0 dB. So for this approach, the theoretical limit is corresponds to the preamplifier with 0 dB noise figure.
Mhm. Okay, but you don't know, I mean, you don't have a number like you're going to do a factor of 10 or 100 times better than
Well,
detection. I mean, even with, so the noise figure generally for a preamp is, you know, a couple of 1 or 2 dB these days.
right.
Mhm.
In this sense, let's say from 1 dB to 0 dB corresponds to enhancement of just 25% and so on. In this sense, you don't find it attractive at all. But the point is, we are fascinated by the idea of mode cooling. Yeah, eventually, when we have optimized the system, we would be able to apply the system to cool the system without lowering the physical temperature, which would change the game, I believe.
Okay. All right, Kong, do you have some questions?
Yeah, just a quick question. I'm just curious for the capacitor that you make. You know, usually, I mean, for NMR we use the ceramic capacitors. They are known to have high Q and low ESR, equivalent circuit resistance. So for the membrane that you know how much is the Q factor or the resistance?
Well,
Roughly.
I've never thought of that before. Well, we don't have any dielectric material. The gap is simply a vacuum, so the loss would be simply determined by the resistive loss of the metal layer, I think. And I didn't care at all about the resistivity of the metal layer. So at this moment, I cannot immediately answer the numerical value or order, but yeah, this is an interesting point. Yeah, let me think about it. Thank you.
Okay, thank you. Yeah, because for us, like, we care a lot about it because like
right.
Yeah, thank you.
So Aaron Rossini has a question. Is there an analogy between optical mechanical detection and the detection of some other high gamma spin for sensitivity enhancement? In other words, if you did, I think he's saying if you did proton detection of C13 or proton detection of nitrogen 15, is there an
I see. High gamma detection means it's a kind of high frequency detection. So in general, frequency detection at higher frequency is beneficial. In this sense, perhaps there is some similarity because the optical signal we use is in frequency around 300 terahertz. And the optical detection itself is done with very, very high sensitivity. So the question is if the signal, if the transduction carries the signal alone, but problem is the intrinsic noise develops in the LC circuit also faithfully transduced to the optical carrier. That's why, yeah, I mentioned that there are two steps transaction from the electrical to mechanical, and then secondary mechanical to optical transduction. And most importantly, we are focusing on the first part because the optical parts can be done with very, very high sensitivity. So it's important to optimize the first stage. I'm not sure if this is a good answer, but there may be an analogy with high frequency detection or
Okay,
high gamma detection.
all right. Okay, so listen, thank you very much, Takeda,
you.
for a really nice presentation. All right.
Thank you very much for
New-
listening.
Interesting new approach to detecting magnetic resonance, all right? Okay, so the second speaker today is Professor Claudio Luchinat from the University of Florence. And Claudio is a very well-known member of our community. He is the co-founder and director of the Center for Magnetic Resonance in Florence, and the co-founder and present director and president of the Inter-University Consortium on Magnetic Resonance of metalloproteins, okay? And so he's going to tell us today about mixed fibrils of A beta amyloids. So Claudio?
Okay, can you hear me?
Yes.
No. So thanks for the invitation. I'm very happy to be able to tell you something about this part of our work. The slide shows where I am from. I think I have to, yes. So this is a magnetic resonance center of the University of Florence, which is also a core center of the European Infrastructure Instruct, which stands for Integrated Structural Biology. So we do a lot of structural biology studies, although we also do other stuff like metabolomics and some kind of biomaterial science and so on. Among the structural biology side, of course, is also looking at the structure of amyloid fibers. And this is the topic of today. And as you all know, solid-state NMR is a good technique to look at the structure, or let's say at the structural features of amyloid fibers. I mean structural features and not exactly structures, because as Bob knows very well, you can call a structure when you have enough restraints for actually fixing the position of the atoms at high resolution. If you don't, at least in the case of amyloid fibers, since you can easily learn what is the secondary structure, then you may need less restraints and still be able to have meaningful structural information. So the center is equipped with, let's see if I can use a laser pointer. Yeah, it's behaving strangely. I don't know if you see a small white square, but if you do, I can just quit this and can you see the arrow?
Yes, yes,
Yeah.
the arrows, yeah.
I will use that. So what I was saying is that you know that NMR has a lot of rivals in terms of structural determination. I mean, the old rival is X-ray structures, of course, and the new rival is cryo-EM. I do think that solid-state NMR, at least in the field of amyloid fibers, has some advantages. And for those of you who do NMR, probably I don't need to explain why. But anyway, we think we get interesting results. So the topic of today is, let's see if I can move this a little bit away. Yes. So back. The topic of today is our relatively recent study where we have shown that A beta 40 and A beta 42 amyloids can actually form mixed fibrils. Now, this was an interesting subject for us, especially because there was a kind of consensus that if you mix together these two type of amyloids, and these are the major components of the amyloids that are generated in the brain, and if they exceed a certain amount, they become toxic, and then they cause neurological disorders. So it's kind of important to look at least the two most abundant, which are A beta 40 and A beta 42. And those two differ just by these two hydrophobic amino acids at the end of the longer one of A beta 42. In the brain, in normal conditions, the ratio between A beta 42 and A beta 40 is about 1:9. So there is an excess of A beta 40. And it's found, it's been found repeatedly that in case of disease, neurotoxicity, we always find that A beta 142 is in larger proportion with respect to 1:9. So maybe 2:9, 3:9, and so on. So this was somehow, this is interesting, which tells you that, well, the simplest explanation is that A beta 42 is more toxic, but this is probably the case, but not necessarily the case. And even so, we actually don't know much about why A beta 42 is more toxic. So in most studies, including NMR studies, but also many other studies, usually the two are investigated separately. So A beta 40 on one side and A beta 42 on the other side. So this was, again, a reason why we got interested in seeing what happens when the two fragments, the two peptides, are mixed together. And just to look at what's been done in the past, and this is not an exhaustive list of the many studies that have been performed, especially by solid-state NMR, on A beta 40, you can see that since 2006, but even before, I think I would start with this work by Tycho and co-workers, 2006, and then 2008, and then also 2008, different structures, because these have the minimum number of restraints that Bob would consider enough to be able to speak of a structural model, if not of a real structure, but the model is accurate enough for our purposes. So these are the topologies. Then when we started working in this field already more than 10 years ago, what we found was that we could get a similar shape of this fibrils, but with a change in register, so that different residues would face different residues on the other side. So this is the N terminal part. There is a loop, and then there is the C-terminal part. And this has been seen many times, including the previous work by Tycho and several other people. So the only difference was that the residues that were facing each other, remember that I should have said that the beta amyloid fibrils, and in most cases fibrils that are neurotoxic, they grow in the third dimension, so you don't see it there, but the growth is in the dimension which is perpendicular to the screen, and it's a normal parallel beta sheet. So what you see here is actually, and some people may actually take this for a beta hairpin. It is not a beta hairpin, because the beta sheet is perpendicular to the screen. So what you have here is interaction among the side chains. And this interaction can be detected by looking at cross peaks in a variety of NMR spectra in the solid state. And so you can see that the pattern of the interactions is different. And you can clearly see that there is a shift of two residues with respect to this structure. Now, let me say something immediately, because I will show structures that differ from one another. This is not to imply that one is good and one is wrong. Absolutely, because there are so many ways of preparing these fibrils that are good and reproducible in each one of them, that we have to cope with the situation that there are multiple solutions to the question, what is the structure of a beta amyloid fiber? And for sure, this is a valid solution. For sure, this is another valid solution. If you look at and some others as well. If you look at the experimental conditions, you find that they are different. We have chosen different conditions with slightly higher pH and agitation, whereas you may use a lower pH and you can keep the protofibrils or the monomers still in solution. So things change and the results can be very different. Now, A-beta 142, this is interesting. It was equally studied a lot, but it was only almost 10 years later that Ishii first and then Bob and Bert Meyer produced, and especially in the case of Bob and Bert Meyer, there were also a lot of restraints. So this is you can call this a high resolution structure. This was not so high resolution, but still you see that the shape is the same and it's an S shape. So it is different, significantly different from A-beta 40. But to me, it is also significant that the solution to the structure of A-beta 142 came about 10 years later, which means that A-beta 42 is not as easy to handle as A-beta 40. It tends to provide faster precipitation of amorphous aggregates. So the conditions in order to get these fibrils must be different. And then and as I said, Bob and others have found these conditions and finally got very good quality fibrils. And then the fibrils touch each other in this way. So the protofibrils make this type of contact between the two protofibrils. And so this is the filament, the mature filament. Now in our hands, the 140 fibrils, which I remind you were these, had this shape. In our hands, the spectra were reasonably good. We had a reasonable amount of restraints, even though there were not as many as in the case of Bob. I want to stress that because I'm sure that Bob will ask the question later. So, but they are more than enough to actually determine the topology with good precision. So the spectra are relatively good. So we could get this structural model in a very confident way. This already happened more than 10 years ago. And those fibrils were actually, at that time, one of the best structural models. Under the same conditions, and I want to stress that you have to compare stuff that is obtained under exactly the same conditions, because if you change conditions, then you get something else, which is equally good, or maybe even better from the physiological point of view, but you actually don't know. So what is important is that your results in your lab and in other labs are reproducible if the experimental conditions are the same. So under exactly the same experimental conditions under which we got the A-beta 40 fibrils, the A-beta 42 provides very ugly spectra. And you see that there are multiple species and there is a lot of heterogeneity, and the fibrils are actually not very well formed. So under those conditions, A-beta 142 doesn't provide good fibrils. In other cases, it does, as you have seen before. But in those conditions, they don't. So what happens if this ratio, the 1:9 ratio, changes? And remember that this changes in people with disease. And it goes invariably towards an increase of the percentage of A-beta 142 with respect to A-beta 140. So this is interesting, intrinsically interesting. And then we got stuck by this paper in 2010 by De Strooper et al. And De Strooper is in Belgium. But De Strooper, he's been studying a lot the formation of fibrils more from the physiological, biological, physiological point of view, not so much from the structural point of view. And so they did a very thorough study, and this is just one of them, where they actually looked at the effect of mixing A-beta 140 and A-beta 142, of mixing them in the same test tube and see what happens in terms of development of THT fluorescence, which is a good indicator of the formation of fibrils, the length of the lag phase before fibrils start to form, and the elongation rate, and so on and so forth. And what they conclude, if they change the ratio from no A-beta 42, only A-beta 40, to 1:9, which is more or less the physiological condition, 2, 2 to 8, 3 to 7, 4 to 6, and so on and so forth, they see that starting from 3 to 7, the behavior indicates, clearly indicates a, let's say, what could be taken as a more toxic behavior, which was also tested with in vivo studies and so on and so forth. So this 7-to-3 ratio triggered our curiosity. And we decided to look at what happens under our conditions. And I stress that if you change conditions, you may get different things. But under our conditions, I'm 100% sure of what we have been seeing. So we started with this 3-to-7 ratio. And the 3-to-7 ratio, since you see this behavior, it would be relatively straightforward to conclude that there must be some kind of interaction among the two fibers, because otherwise you would not explain this phenomenon. Because at this point, if the toxicity was simply due to the increase of a beta 142, then you would see still an increase in toxicity by increasing a beta 142, whereas it seems to stop at this 3-to-7 ratio. So the obvious idea, at least the idea that the Struver and others had, was that, well, there must be some kind of interaction among these two different peptides. But then Sarah Lince and others, and I want to stress that this was not the only paper, reporting segregation of A beta 40 and A beta 42. And segregation is described by them as if you put together a beta 140 and a beta 42, they self-assemble into separate homomolecular fibers in binary mixtures. So they actually don't know each other. They separate, they segregate. However, in that same paper, the description is that you may have primary nuclei of only a beta 42, only a beta 40, but also mixed nucleation sites. So the finding was explained that although the mature fibers were actually only a beta 40 or a beta 42, that monomeric A beta 142 accelerates the aggregation of A beta 40 in a concentration dependent manner. So there is an interaction, which does not seem to be manifested in the mature fibers. So this was even more interesting to us. So we started our own work with solid state NMR by looking at mixtures of A beta 42, A beta 40 under the same conditions as we knew we got good fibers of A beta 40, but not good fibers of A beta 142. And we started from this 3-to-7 mixture, which was the one that the Struper considered the most interesting ratio. Now, you have to follow this, this slide because I will put additional layers here. So we start from the black spectra, which are the mixture of A beta 142, A beta 40 in 3-to-7 ratio. And you can tell from the spectra, if you look at them carefully, that there are more cross peaks than you would expect for a single species. And this is clearly confirmed if you superimpose the A beta 140 spectrum to the spectrum of the mixture. You see very clearly, and I can switch back and forth, that there are some signals in the black spectrum that are absolutely superimposable with the signals of A beta 140. Actually, you could find a signal in the mixture that was corresponding strictly with a signal of A beta 140. But then there are residual signals that are not covered by the blue, and the same here. So the conclusion is that we have two species here, one of which is pure A beta 140 and is the same, exactly the same A beta 140 fiber that we had characterized 10 years before. What is the other species then? Well, the other species, you can guess what it is if you start again from the 3-7 and superimpose to that another mixture where this time you only have 142 and 140 in 1-to-1 ratio. So we said, okay, if there is an an excess of A beta 40, and we do see the spectrum of A beta 40 coming up, well, the conclusion should be that A beta 40 indeed segregates, if you want to use the word, as it was found by Sarah Lindsay. But then what is left is a mixture, is a one-to-one mixture of A beta 40 and A beta 42 that apparently provide a single species. Now, what is this single species? You can see here that the red spectrum, which is superimposable to the black spectrum, is actually kind of complementary to the spectrum that we had seen before. So what is not accounted for by the presence of pure A beta 40 is accounted for by a mixture of A beta 40, A beta 42. I should stress that in all of these experiments, only A beta 140 was isotopically enriched in carbon and nitrogen. Okay, so this is interesting because we can conclude already at this point that we have two species when we are in the 3:7 ratio, one of which is pure A beta 40, and the other one is a one-to-one mixture of A beta 40 and A beta 42. And this one-to-one mixture, you only look at the mixture from the point of view of A beta 40. This one-to-one mixture has a spectrum and a structure, because we have enough restraints that we can tell the structure, that is similar but not the same as A beta 40. So, okay, here you can do the same by actually still enriching A beta 40 and non-enriching A beta 142, carbon nitrogen, and you can tell that everything is confirmed. So we do have a species, which is a single species, that is an equimolar mixture of A beta 40 and A beta 42. And we also have the sequential assignment. And from the sequential assignment and the cross-peaks, we can easily see that we have the usual pattern of beta probability that tells you that you have a short beta at the N terminal, which is not connected to the rest. And then you have beta 1 and beta 2, which are connected to one another. And they are connected in this way. And I will get to this a little later. And so they can form a protofilament, which is the same as we have seen and as Tycho had seen. But this time, the protofilament is made of a one-to-one mixture of A beta 40 and A beta 42. And we are only monitoring A beta 40. Now we look at the details, and we have again a reasonably good number of restraints that allows us to tell that the registry between these two beta strands is different from what was seen in pure A beta 40 by us. So we are looking at A beta 40 in a one-to-one mixture with A beta 40, A beta 42. It's a C-shaped fibril, or protofibril, protofilament if you want, but the register is different. And guess what? The register is the same as the one that was observed by Tycho on A beta 40, which I call here alternate A beta 140 structure. So under different conditions, A beta 140 has this type of shape. And also there was an early A beta 142 structure with actually not enough restraints to be able to tell much, but Smith and co-workers, long before the beautiful results of Bob and Bettmeyer and Ishii, that was showing this same type of arrangement. So what happens to the other filament? Because as you may remember, the other filament is beta 2, beta 2. And this makes the complete filament. So beta 2 is facing each other in the same way as we have reported for pure A beta 40. So this new filament made of two protofilaments is very similar to the one of A beta 40, but is also similar to some findings for early findings of A beta 42, and also findings by Tycho on A beta 40 itself. Then you have a very similar structure, but it's not identical to the one that we had with pure A beta 40 under these same experimental conditions. So this is the protofilament, and this is the complete filament, which is arranged like this behind. Okay. So how is it made? So we know it's 1-to-1, and there is no doubt. We have repeated, I haven't shown that, but we have repeated the same experiments by enriching A beta 42. So we know that we are getting a single species when we mix A beta 40, A beta 42 in 1-to-1 ratio. And what we see, the possible results are that you get two protofilaments, one of which is A beta 40 and the other one is A beta 42. This is possible. We didn't like this explanation because this would be prone to not getting stoichiometric relationships. If A beta 40 is allowed to make fibrils by itself, it makes the other kind of fibrils. So why should it face A beta 42? And what would be the structure of A beta 42? Remember that we have only one species, and the one species looks pretty much the same if you look at it from the side of A beta 40 or from the side of A beta 42. So the other, to us, more reasonable explanation was that we could have an interleaved situation where there are one strand of A beta 40, one of A beta 42, and so on and so forth. And the interleaved situation could actually be of two types, but we really are not able, and we have not been able to distinguish between these two. But we can actually distinguish these two, either of the two, from this one. And this is something that we have done in the same work by doing these two types of experiments that I'm going to describe in a little detail. So this is an HHC, where actually you see cross peaks between nitrogens and carbon, and you see cross peaks in the carbonyl region and cross peaks in the C alpha and mostly C alpha region. Now, how is this sample made? Is a 1-to-1 mixture of A beta 42, which is 59 enriched, and A beta 40, which is C13 enriched. Now, if the situation would be of two protofilaments, one of A beta 40 and the other one of A beta 42, besides the fact that this would not explain why we get something which is different from the structure of A beta 40, but then you should not have any cross peak or very, very weak cross peaks, because the two protofilaments would be distant from one another. And moreover, the strong cross peaks that you get between the nitrogen and carbonyl carbon are typical of the hydrogen bonds that keep together the strands along the elongation axis. Well, an obvious proof is that if you just look at nitrogen 15 A beta 142 with A beta 140, which is not enriched, you see nothing. So there is no way to interpret this as a weak response from the carbon 12, the 1% C13, which is in the natural abundance. This is a real spectrum where C13 enriched material is actually close to nitrogen 15 enriched material. So this to us was already a sufficient proof. A referee was asking us to do also a Tidor experiment, which was actually developed by Bob some 20 years ago, I should say. And in the Tidor experiment, you see the same thing. You see that if you enrich by nitrogen 15 the A beta 42 and by C13 the A beta 40, you do see a significantly strong signal of the carbonyl and some signals in the C beta regions. If you only have nitrogen 15, you see nothing. And if you have double enriched, so double enriched is interesting because this is a spectrum which potentially can give you the same strength of this signal. This signal is almost not present. So this signal can only come from the direct contact of A beta 40 and A beta 42 when they alternate. I think this is the final result. Only an interlaced structure is in agreement with the experimental data. So what does this mean? Does it have any physiological or pathological relevance? Yeah, of course we don't know. But we don't know what is the relevance of the mature fibrils of A beta 40 alone or of mature fibrils of A beta 42 alone. So this is just another brick in the wall, if you want a quotation, in our long process of trying to understand the formation of fibrils. One lesson that you have certainly learned is that what you get is strongly dependent on the experimental conditions. If you are careful and if you use the same experimental conditions, you get the same results. So these data are reproducible, these results are reproducible. Other results obtained in different conditions may provide different answers. So just going back to the paper by Sarah Vince and others, it could have been it's perfectly okay that in those cases, there is no formation of mixed fibrils, but just segregation of A beta 40 and A beta 42. By the way, by looking back in the literature, this was an EPR study with radicals, with spin labels, where people had proposed that indeed A beta 42 and A beta 40 could be interlaced. But of course, this is in the language of Bob, this is a one restraint structure. So I think this is a little bit not enough to say, but it's interesting that this proposal was actually made before the paper by Sarah and before, of course, our paper. Conclusions: A beta 42 and A beta 40 in experimental conditions can co-fibrilize and form a one-to-one interlaced fiber. And this is for sure. A beta 142 and A beta 40 in the mixed fibrils have a U-shaped arrangement that differs from both the U-shaped conformation observed in fibrils of pure A beta 40 and the S-shaped conformation of fibrils in pure A beta 142. Again, are the other results less reliable? No, absolutely not. They are equally reliable. And all results in this field are reliable as long as they are reproducible under the same experimental conditions. Mixed fibrils preferentially form even when A beta 42 and A beta 40 peptide are mixed in a non-stoichiometric ratio. So now this is a point that I would like to stress because allowed me to make some kind of speculations that are purely speculations. So you are free to not believe in what I'm saying. But if you mix A beta 40 and A beta 42 in a ratio which is different from 1 to 1, and if you have an excess of A beta 40, which is what happens in healthy people, a large excess of A beta 40, there is nothing wrong in believing that A beta 40 can make fibrils by itself. So in a way, it segregates. A beta 42 is a small amount, and maybe it's below the threshold above which fibrillation can start for A beta 42 as well. By the time you increase the amount of A beta 42, you start form mixed fibrils. And if you form mixed fibrils, it would be tempting to speculate that this is a defense mechanism by the organism. If for some reason there is an imbalance and if A beta 42 starts getting more than it should be, then at least at the beginning, A beta 40 is able to sequester this A beta 42, forming these mixed fibrils. Beyond a certain threshold, which could well be 3 to 7, then we might get such an amount of A beta 42 that A beta 40 is not good enough to prevent a selfish behavior of A beta 42, which would bring to formation of toxic species, maybe not even fibrils, maybe small aggregates, protofibrils, or whatever. People think that maybe the more toxic species before getting to the mature fibrils. And I think I will stop here. These are the people who have contributed to the work. The first three, Marco, Enrico, and Linda, have been the major players, and also Sara, Domenico, Magdalena, Gianluca, and Leonardo. Over a few years, they have contributed a lot. And then Bardo Struper and Irina Benilova, with whom we have published this paper, and Eric Wenker and co-workers, who's also a co-author of this paper. And Eric was with us in IRANET Neuron, which was a European program by which we got a little funding to study this type of amyloid fibrils. And with this, I thank you for your attention.
Hey, well, thank you very much, Claudio, for that really nice presentation. Let me turn my camera back on. It won't turn. There we go.
It was on.
It's a little bit slow. Yeah. So I mean, it looks like you have really found one-to-one fibrils here, intercalated one-to-one fibrils. can I ask, how many times have you done that experiment?
Now the 2D, we did it twice on different preparations. The Tiber we did only once.
Uh-huh. Uh-huh. Yeah.
But then we have all the other experiments of the those were also made more than once, of the overlap of one enriched, the other non-enriched, the carbon-carbon and normal carbon-carbon, DAR and whatever.
Mm-hmm. Mm-hmm.
So yes, it is reproducible. I mean, we haven't done it 12 times, but it's been done more than once.
Uh-huh. Okay. Well, I mean, it's in certain ways, it's not unreasonable that, I mean, the amino acid sequence of the two peptides is very, very similar.
It's the same except two. Yeah.
Yeah. So, I mean, except for the IA at the C terminus, it's not at all unreasonable that this kind of thing could happen. I guess it just has not been seen previously, though. Yeah, by other people. I mean, I'm not sure I have many questions. You give a very, very clear talk here on this, and it's certainly true that when people change conditions a little bit, they can change the quality and quantity of the sample and the fibrils that they get. All right, and that is a concerning problem with the whole field of amyloid, NMR, and so forth. All right. So, Kong, do you have some questions for Lucio? Claudio?
Not at the moment.
I can, if you want, I can try to answer a question that you might have asked, but you haven't.
How many constraints did you have?
No, this, no, I know. It's about a total of 40 something, which is not as many as you have, but it's enough to determine the register. But the question I thought you might ask, and again, it's a speculation if you want from my side, but I like to think that a beta 42 is less prone to assume this C shape because of those two hydrophobic residues. Because if you look at the model of the C shape, those two hydrophobic residues, which are rather bulky, would somehow bump into each other, and so make the match at the end of the sequence less perfect than you would like to see, which is not the case in the S shape, where you get a better situation. But if you mix it with the beta 40 and you get this interleaved fibers, then you can imagine that at the end of the C terminal of the peptide, you have once you have a beta 40, then you have a beta 42, then a beta 40 again. So there's more room for the last two residues because they are interleaved with a beta 40 with this, which is two residues shorter. That might be an explanation. And the other thing I wanted to add about the conditions, which I haven't stressed probably enough, was that our conditions were pH 8, which is a little higher than most people use, and agitation. And agitation to us, and I also heard some remarks about this from the seminar of Rob Tico yesterday, that agitation may actually help get into the right thermodynamic well, which is by no means more physiological. It can be actually away from physiology, but it tends to provide fibers that are the most stable. Whereas if you let it go without agitation, you may get forms that can, with time or with subsequent agitation, be destroyed and reformed into a thermodynamic, more stable product. But this is again speculation.
Okay. So Christian Grayson, you're raised a question. You may have sort of answered it already. It says, I may have missed it, but are the last two amino acids of 1 to 42 included in the amyloid? He means in the amyloid core.
Well,
go back to your slide. I don't know if you
let's see. Oops. Well, yes. I mean this tail here, and you see the tail is is shorter for A beta 40 and longer for A beta 42. This is not a very good representation, I admit, but if you want, you can look at this. The answer is, I think, that we do see the signals for those from those two residues, which means that they are ordered. Now, their order doesn't necessarily mean that they are perfectly bound in the, because being longer, they may not participate in the beta structure, but they are ordered. I don't know if Christian is happy with this explanation, but this is what we see. We see the signals and they're clear.
Christian, if you want, do you have other questions?
Thanks for including me. Now I was just thinking of what might be the, can you hear me?
Yes.
Yes.
I was just thinking what might be the driving force for this mixture, and one could of course be that maybe the negative charge of the C-terminal carboxylate or so, by that has a bigger distance or so than of the beta 140, and that this is kind of a buffer also to make a bigger distance of this carboxylate. I'm just wondering why a 1-to-1 mixture might be thermodynamically more favorable than the because I mean in a way entropically or so I mean some random distribution might be more likely than exactly 1-to-1 with alternating incorporation of the peptides.
Well, I see the point. Of course, there is no satisfactory answer, but we have seen also in other cases that if you use our conditions, which is agitation, relatively strong agitation for a long time, we tend to get, we get always the same species if we work under the same conditions. And the species is stable in the sense that if you leave it there for months or it doesn't evolve anymore, which doesn't mean that this is a real thermodynamic minimum, but at least is stable. Now, why I think that this may be more stable than pure A beta 40, A beta 42? Well, pure A beta 40 is very stable. A beta 42, as we know, tends to more easily provide aggregates that are not well shaped, the fibers that are not well formed, except when you find the right conditions, as it was the case for Bob, for Bert Meyer and Vichy. The question is whether the mixture is even more stable than A beta 40. Well, if you mix them in different amounts, and we have done 7:3, we have done also another ratio, you always get the excess A beta 40 by itself, and what is left is a single species. So to me, this means that as long as there are A beta 40 and A beta 42 present, they preferentially make fibrils together. And then what is left is A beta 40. So again, by wild speculation, you might say that these mixed fibrils are more stable than A beta 40 by itself, and even more stable than A beta 42. Now, I was trying to offer a very naive explanation for that by saying that the two hydrophobic residues at the end, which are the ones that probably prevent A beta 42 to adopt the C-shaped form that is seen easily for A beta 40, those two residues could be actually diluted by interleaving A beta 40 and A beta 42. If you do not have this interleaved situation, then you would have strands where you have more than one A beta 40, A beta 42 together, as well as some A beta 40 together. And those would be less favorable. So that's why we think that it's really interleaved. And the NMR data, two different types of NMR data seem to point, or actually clearly point to this, because you do see a strong signal which can only come from coupling between the nitrogen 15 on one peptide and the C13, the carbonyl C13 of the other peptide. Again, I don't know if you expect something.
I have to digest.
You don't expect a proof, right? Because we don't have a proof for that. But it may be a reasonable. It may be a reasonable explanation.
Okay, thanks a lot.
You know, the structure that you're suggesting is, well, perhaps you're familiar with this cryo-EM thing, cryo-EM structure that just came out from studies of brain from the Cambridge, from the Goddard and Shores group. Yeah.
I see. Yeah.
And that has the sort of, it has an S, more or less S-shaped. It's a different S than what we found with 1 to 42. Now, if you look in the supplementary information of that paper, you'll find that the fibrils were actually, they weren't pure by any means, that they were a combination. There was 1 to 40 in it, there was 2 to 42, 8 to 42, 4 to 42, pyroglutamic, 4 to 42. There was a mixture of different types of A beta present. And these were brain samples extracted from brains. And there were basically a few samples, 2 or 3, but they came up with 2 different structures and both had the S shape. They claim for the 1 to 42, okay? Now that doesn't, you know, I'm just saying this is what supposedly happens in vivo. And we don't know if, I mean, this was a few brain samples. I mean, if you had more brain samples, you might be able to say this was really a generally applicable structure. Christian, maybe you have a comment on that? You still there?
I did not see the structure you were just referring to, so I can't.
Well, it was a paper in Science. Yeah. Yeah.
A very recent one, and it's indeed it's S-shaped. It's more than one form. Well, I mean, it's not homogeneous, but I mean, it comes from brains, so you can also imagine that it's not homogeneous.
Yeah.
The fact that it is S-shaped points towards this type of fibril for A beta 42 being more favorable than the C shape, and I think there is little doubt at this point.
Yeah.
But then the question, if I can move the question backwards, what's before that? I mean, what are the small aggregates that seed the formation of fibrils? And the question that, I mean, I was trying to provide not an explanation, but food for thought, if you want. When in a brain of a healthy person, you start seeing an increase of A beta 42. It's known that people don't get sick immediately. There is usually a number of years before the manifestation of the disease. And at that point, usually the amount of A beta 42 is in fact much higher. Much higher means like 30% instead of 10% of the total. So if you couple this observation, which is clinical, with the observation that, well, that mature fibrils may not be the cause of the disease, but could be actually a way for the organism to slow down the disease, to keep it from getting worse, at least to a certain extent, then these mixed fibrils might actually be a way for people to get sick later, because as long as you have an excess of A beta 40, you might actually find that A beta 42 prefers to make this type of mixed fibrils with A beta 40 before actually giving rise to the toxic species that may not be mature fibrils, but may be very short fragments of A beta 42, which is known to be, well, I hate to say A beta 42 is more toxic because we don't know, but let's say to be associated with higher toxicity.
Yeah. Okay. All right. Well, thank you for a really nice, very interesting talk. inspiring. You've asked as many questions as you've answered, I think, today. But it's really quite a nice new result, and it's something that has not really been seen previously in the A beta community. So again, Claudio, thank you very much for your talk, and I would like to thank Takeda. It was 12 a.m. for him, so he adjourned and went to sleep after his presentation, but he gave a very nice presentation as well. And Kong, next week, who do we have on, or 2 weeks from now, who do we have on?
Yeah, 2 weeks from now, we will be hosting Anja Bokman and Patrick Van der Velde.
Yeah, so Anja Bachmann from Lyon, CNRS in Lyon, and Patrick Van der Velde from Groningen University in the Netherlands will be here to talk. Okay, so thank you very much again, everyone, and we hope to see you in 2 weeks or so. All right, bye.
Bye.